Hello guys, good day. I am Franzelle Janagap, studying at University of San Agustin, taking up Bachelor of Music, major in Music Education. So for today's vlog, I and my fellow teammates are going to talk about our Module 11 in the course of Science, Technology, and Society, which is entitled Gene Therapy. But before any of that, my fellow teammates will introduce themselves first.
Hi, I am Demmie Nicollete G. Maza, and I am taking up Bachelor of Music in Music Education.
Hi there, I'm Tiffany Rose Montinola, taking up Nutrition and Dietetics.
Hello everyone, I'm Kayezia Garcia, taking up the Bachelor of Science, Nutrition and Dietetics.
Hello guys, my name is Bea Joliah Liego. I'm from BSND3. So the question is, what is gene therapy and cell therapy? Gene therapy is an experimental technique that uses genes to prevent or treat the diseases. So the cell therapy is uses cells for the same purpose. So both gene and cell therapies have potential to treat the underlying cause of both genetic and acquired diseases by replacing the missing proteins or cells.
How does gene therapy works? Gene therapy works by changing the genetic information of a population of cells for the purpose of alleviating or combating the cause or symptoms of a disease. Some types of viruses, when used as a vector, incorporate the genes in the chromosome of the host, as what happens in the retroviruses. However, when adenovirus is used, there is no integration of the donated gene to the chromosome of the host cell. In any case, both vectors can deliver the goods, but there are many challenges in this technique. Firstly, this technique is largely experimental and therefore far from perfection. Second, they have to make sure that the newly introduced gene actually works by synthesizing the desired protein. In simple terms, gene therapy works by replacing a defective gene and changing the genetic information of cells for the purpose of prevention of a disorder. Gene therapy has been attempted on somatic body cells for diseases such as cystic fibrosis, cancer, and severe combined immune deficiency syndrome or SCID. The most common form of gene therapy is germline gene therapy, which involves modifying the genes in egg or sperm cells, which will then pass any genetic changes to future generations.
What are the strategies used in gene therapy? The strategies used in gene therapy are gene addition, gene correction, gene silencing, reprogramming, and cell elimination. So these strategies are mainly used to target specific population of somatic cells.
What are the uses of gene therapy? First, replace missing or defective genes. Second, hasten the speed of destruction of cancer cells. Third, supply genes that cause cancer cells to revert back to normal cells. Fourth, deliver bacteria or viral genes as a form of vaccination. Fifth, provide genes that promote or impede the growth of new tissue. And sixth, deliver genes that stimulate the healing of damaged tissue.
What are the common types of cell therapy? The most common type of cell therapy is blood transfusion, which is a transfusion of red blood cells, white blood cells, and platelets from a donor. Another common cell therapy is the transplantation of hematopoietic stem cells to create bone marrow, which has been performed for over 40 years.
The question is, how are viruses used in gene therapy? Viruses are used as gene delivery vectors. In order to deliver the genes to the cell, only genetically engineered vectors such as virus are used in gene therapy. These newly engineered viruses protects the new gene it carries from degrading enzymes in the blood. The virus, being an obligate intracellular organism, forces the cell to take up the new gene. and goes into the nucleus. Once into the nucleus, or integrated into chromosomes, the new gene starts to perform its new function to synthesize a new improved or functional protein. However, there must be a room for a new gene. To summarize my answer, viruses are used in gene therapy as gene delivery vectors as oncolytic viruses.
Can viral vectors make you sick? Viral vectors and oncolytic viruses are designed to reduce the risk of adverse effects. However, every medicine has risks. Possible risks are the possibly prolonging or worsening symptoms of complicating the condition with adverse effects of the therapy, the expression of the genetic material, or the survival of the stem cells may be inadequate and or may be too short-lived to fully heal or improve the disease. An example of a viral vector is retroviral vectors. Retroviral vectors are commonly used and known to integrate into the genome of the infected cell in a stable and permanent fashion. Reverse transcriptase in the virus allows integration to the host of the genome. There are two types of retroviral vectors, the replication competent and replication defective. Usually, replication defective vectors are preferred in practice as they allow for several rounds of replication due to their coding regions. Another example is the adenoviral vectors. Adenoviral vectors have a wide range of action and are able to deliver nucleic acids to both dividing and non-dividing cells. Adenoviruses are often responsible for the respiratory and gastrointestinal and eye infection that affect humans. As a result, research is currently being conducted to investigate the use of adenoviral vectors in applications of gene therapy and vaccination. In conclusion, viral vectors cannot and will not make us sick because these viruses are used in gene therapy and it is designed to reduce the risk of adverse effects. These viral vectors are tested in animal and in human trials to make sure that human cells are not harmed. However, like all treatments or therapy, it has its risks. Therefore, anyone availing this therapy must discuss with their physician and other health advocates the pros and cons of gene therapy.
The characteristics of diseases that can be treated by gene and cell therapy are those for which there is no effective treatment, those with a known cause or a defective gene, and those that have failed to improve or have become resistant to conventional therapy. The three types of diseases for gene therapy can be distinguished in monogenic disorders, polygenic disorders, and infectious diseases. Monogenic disorders are single locus or gene which is defective and responsible for the disease. It is 100% heritable, which means that we acquired this disorder from our ancestry. The examples are sickle cell anemia, severe combined immunodeficiency, and many more. On the other hand, polygenic disorders have multiple genes involved. These diseases may be dependent on environmental factors and lifestyle. Examples are heart disease, cancer, diabetes, and Alzheimer's disease. The severe out of the three is the infectious diseases. These diseases were caused by living organisms like viruses and bacteria. They can be passed from person to person through body secretions, insects, or other means. So the examples are HIV, tuberculosis or TB, and coronavirus or COVID-19. What is the future of gene therapy? This technique may allow doctors to treat a disorder instead of using drugs or surgery. Its accuracy can eliminate cancer cells without damaging normal and healthy tissues. So that's all for today's vlog. I hope that everyone have learned a lot about gene therapy, its pros and cons, and the future of having this gene therapy.